to take their seats, please, and we'll start the next session maybe in a minute or so. Good morning. I'm Dheeraj Yadav from the University of Pittsburgh, and my co-moderator is Dr. Darwin Conwell from the University of Kentucky. We are excited to start this next session on endocrine and exocrine pancreatitis crosstalk. We have 4 excellent speakers. We would take the questions for the whole session at the end. Our first speaker for the morning is going to be Dr. Phil Hart, who's a professor of medicine in the Division of Gastroenterology and Hepatology at Ohio State University. Phil has been leading many national initiatives in the area of diabetes in the setting of pancreatitis. He's the PI, co-PI of the DETECT study for the CPDPC. And he's also co-lead for the longitudinal observational study, DREAM, which is assessing diabetes after acute pancreatitis. Phil is going to be talking to us about bone health and nutritional deficiencies in pancreatitis.
Great, thanks for the introduction. I'd like to thank the meeting organizers for this invitation, and thank you for being here. My daughter is with us at the hotel, and this morning she told me that if I make any mistakes to just keep going. And so I came up the stairs earlier, tripped, spilled my tea, got a refill, and here I am. So in the next 10 to 12 minutes, I'll be talking about 2 different topics that affect many of our patients with chronic pancreatitis that we often don't talk about or even think about for that matter. These are my potential disclosures, although none of these are relevant for today's topics. So my objectives are simple. First, to review the contributing factors and some aspects related to management for osteopathy and chronic pancreatitis, and then to do the same for micronutrient deficiencies. So to begin, we start with the question, well, how common is osteopathy in chronic pancreatitis? And it's quite interesting that whether it's single center studies, multi-center studies, Europe, North America, the point prevalence estimates are very similar and consistently high for both osteopenia and osteoporosis, as shown here. Why does that matter? Well, the end clinical outcome that we're trying to avoid in these patients is that of fracture, particularly low trauma or spontaneous fractures. And in a study that Dr. Conwell had previously published, they demonstrated that the odds of fracture in our patients with chronic pancreatitis is significantly higher than in healthy controls from the general population. And you'll also see too, these estimates are actually higher than what we see in patients with inflammatory bowel disease, which is noted to be a high-risk population. Well, what are some of the contributing factors behind why we see these high prevalence rates? Well, first off, there are shared risk factors between both osteoporosis and chronic pancreatitis, including cigarette smoking and heavy alcohol use. But there are also some more specific or unique contributing elements from chronic pancreatitis, particularly the systemic inflammation that we see in our patients, but also vitamin D deficiency, which we'll come back to. Now, I don't have time to delve into all of the different aspects of it. And in fact, the mechanisms and the connection between systemic inflammation and osteopathy and chronic pancreatitis hasn't been worked out. However, when we look at these connections in other diseases, it's been well established that systemic inflammation through a variety of mechanisms, including the rank ligand pathway, can generate an imbalance between osteoblast and osteoclast activity that favors bone resorption. And in really great work that Sinead Dugan has published previously, she was able to demonstrate that systemic inflammation is associated with increased markers of bone turnover, which is demonstrated in the figure to the right. Now, when we look at our patient population, for other potential risk factors, there are some that stand out consistently, including older age, female sex, and lower body weight as estimated by the body mass index. So these again are not surprising. These are common risk factors for osteoporosis or osteopathy in the general population. But one of the things that I think is really remarkable in data that we recently published from the PROCEED multi-center study is that the rates of osteopathy remain high even in the groups that you wouldn't expect to. And I put red bars around to show that in older men, the rates of osteopathy were over 50%. And also in premenopausal women, again, the rates were in some age categories as high as 50%. So there's definitely a contribution from age, sex, and body weight. However, there's still something going on in our patients that's accelerating this process. Now, one interesting thing that does not stand out are various characteristics of the disease, at least those crude characteristics that we often phenotype, such as calcifications, pancreatic atrophy, duct dilation. There didn't seem to be any connections between these crude elements of the disease characteristics. Well, so it's a common problem. So this begs the question, should we be screening for osteoporosis? I think the answer is pretty clear. Yes, we should be doing this. There are a number of groups, societies, and statements that have emerged from multidisciplinary meetings that suggest that, yes, we should be doing this. However, the other way to ask that question is, are we doing this? And the answer to that is, I think, if we're all honest, probably not, or more definitively a no, shown in a single center study at a academic medical center, where only a fraction of patients with chronic pancreatitis had ever had imaging for osteoporosis. Now, I'll also say in the PROCEED study, which I alluded to previously, we actually offer all patients a free DEXA scan. So this really mitigates or eliminates the potential concern to say, well, it's not covered by insurance, patients don't want to pay for it. So we eliminated that barrier, and only 50% of patients, or roughly 50%, took a DEXA scan. So there's more work to be done here. And so the question is there any way that we can shortcut this or work around this? I think possibly. So this is something that's been described in general population studies that's referred to as opportunistic screening. And specifically what the idea is taking bone density estimates from CT images and using those as a surrogate for the T-scores, which is sort of the T-scores from a DEXA scan, which is the gold reference standard for osteoporosis. And in this large study, you can see there's a very good linear correlation between bone density as estimated by CT images of a lumbar vertebra and the T-score from DEXA scans. So this led to a pilot study that was led by Julia McNabb, where we looked at a small number of patients with chronic pancreatitis. And obviously the estimates are not as precise considering the sample size, but we do see correlation, suggesting that this may be a future opportunity for us to pursue further. So osteopathy is common in our patients. There are multiple different factors that can contribute to this. We need to learn more about the mechanism, specifically of how inflammation contributes to this. And we also need to do more work to think about and understand how we can implement the recommendations better. So I'll shift gears to talk about micronutrient deficiencies, and I'll interrupt myself before I even ask the question of how common are they to just not make any assumptions and answer the question, what is a micronutrient? So I think if I'll be honest here, that I've often thought micronutrient equals fat soluble vitamin, but it's actually more broad than that. So macronutrients and micronutrients are held in distinction to one another. Macronutrients are the primary source of energy in the food that we eat. These are fats, proteins, carbohydrates. Micronutrients are basically everything else. These are items, vitamins and minerals, that is, that are consumed in much smaller and sometimes even trace amounts. Okay. So back to our question, how common are micronutrient deficiencies in chronic pancreatitis? Well, we've really, as a field, primarily focused on the fat-soluble vitamin deficiencies, which some estimates are shown here from a couple different studies. Some other, mineral deficiencies that have been looked at, magnesium and zinc, also, fairly or relatively common. But really beyond that, we don't have great studies that have been done in a systematic manner. But if we zoom out and just think about what is the nutritional status of our patients, you can see here the estimates are that over a third are underweight or have a low BMI. And again, there's a wide range, but many of our patients have sarcopenia. So this is clearly a high-risk nutritional population. And we're really, with what we understand about micronutrient deficiencies, we're probably just seeing the tip of the iceberg. Well, this ends up being a complicated issue to understand because there are multiple potential factors that can contribute to the development of these deficiencies in our patients, which include decreased intake, which in turn has multiple potential factors that can relate to it. Poor diet quality, and previous work that we've done, we've shown that not only are there challenges with just the overall caloric consumption for many patients, the quality or the composition of the nutrients that patients are taking in is often poor. Third, there's the issue of fat avoidance, malabsorption, particularly in the setting of EPI. And then lastly, again, back to the inflammation problem, is that chronic inflammation, even at a low-grade level, can disrupt the binding affinity of nutrients and proteins and also alter protein levels. So this is a couple images from a patient of mine in clinic who had a rash for several months, treated all different sorts of ways. One of our dermatologists looked at this and was able to pinpoint that this was a type of rash called necrolytic acral erythema, which occurs in the setting of zinc deficiency. And so this patient started on zinc supplementation and very quickly experienced improvement, just to illustrate, one of the many micronutrient deficiencies that can pop up. Well, what should we be doing for these patients and what does the evidence tell us? Well, one of the challenges is that there are very few data to really, number one, characterize the problem, and number two, therefore, to guide recommendations. So any of the statements that have been published are really heavily influenced by opinion. And again, considering the lack of certainty in the data, the recommendations are very, I think appropriately vague and sometimes soft. Nevertheless, the recommendations have been put forward from multiple multidisciplinary meetings and societies, and I'd refer you to 4 of the more recent groups who have talked about this. But just to avoid leaving sort of a complete ambiguity, I like to be concrete, and so just share at least my practice and how I think about nutrition screening in patients with chronic pancreatitis. So I think that there should probably be some what I call untargeted initial sieve that, whether it's nurse administered or something quick that the healthcare provider can administer. I like the MST, which is a very simple 2-part questionnaire that our nurses in clinic administer. For patients who are screening positive, then diving more deeply into this with either a formal or an informal nutrition or dietitian consultation, or in a very comprehensive dietary intake method such as a FFQ. I prefer to screen on an annual basis, irrespective of nutritional risk assessment for the micronutrients that are shown here, as well as with a C-reactive protein, again, to understand whether or not some of the low levels may just be falsely low. And then lastly, as an overarching statement, I just have a very low threshold to include our dietitian team. So in summary, osteopathy is a common complication for individuals with chronic pancreatitis. Micronutrient deficiencies, as I mentioned, have multiple potential contributing pathways, but are also common. With that said, these estimates are imprecise and can vary from study to study. I think that additional studies are needed to help us understand the mechanisms of these different nutritional sequelae. And then lastly, I think that further investigations are needed to help us generate the evidence base such that we can provide our patients with nutrition-related recommendations. So with that, I'd like to acknowledge my collaborators at Ohio State and sources of funding, and thank you for your attention.
Good morning. Thank you, Phil. That was excellent. Our next speaker is going to be, it's a pre-recorded lecture by Dr. Mark Gardarzi, who's the director of the Division of Endocrinology, Diabetes, and Metabolism, and a professor of medicine at Cedars. Mark is the Eric M. Field Chair in Diabetes Research. We've all got to know him even more in his leadership in the T1D APC, where he co-chairs the genetics interest group, and also in the CPDPC, where he co-chairs the type 3C working group. It's exciting for us to see this crosstalk. I mean, that's why this session is put together. We all know that we need to be talking more to our endocrinologists. You'll hear from Melina later, who's always one of our advocates. But this is great to hear Mark talk about this. He's a leader in the field, and it's very exciting research that he's going to be talking about. So let's hear from Dr. Goodarzi. It's pre-recorded, as I said.
I'd like to thank the organizers for giving me this opportunity to speak to you today on the crosstalk between pancreatitis and diabetes. This is a study out of the UK, which followed 32,000 individuals with new onset diabetes as adults. And this is a good reflection of the proportion of diabetes that we see. 97% were diagnosed with type 2 diabetes. About 2% had diabetes occurring after pancreatic disease. And they subdivided this as after acute pancreatitis or after chronic pancreatitis disease, which is mainly chronic pancreatitis. And this was more common than type 1 diabetes, which accounted for about 1%. They followed these individuals and found dramatically that those with pancreatic disease, diabetes, required insulin at a much more rapid rate. So you see the blue line for type 2 diabetes, 5 years out, only 4% of those patients needed insulin. However, pancreatogenic diabetes, overall, 30% needed insulin at 5 years. And you see for chronic pancreatic disease related diabetes, 40 to 50% required insulin. The study also found that most of those patients with post-pancreatitis diabetes were not recognized as such, only 3%. The majority of them were actually diagnosed as type 2 diabetes. Another thing that they found was that those with pancreatogenic diabetes had higher hemoglobin A1C levels, worse glycemic control versus type 2 diabetes. A separate study found that CP-related diabetes comes compared to type 2 diabetes as a much higher risk of severe hypoglycemia requiring assistance. So you can see right away, it's important not to miss this diagnosis. The worse glycemic control, the higher rates of hypoglycemia, so it's really important to pick this form of diabetes up. It requires closer monitoring, quicker advancement of treatment, and treatment of the chronic pancreatitis if symptomatic. So anyone who sees a patient with diabetes, at least on the initial evaluation, should search for a history of pancreatic disease. Other reasons why it's important not to miss the diagnosis. CP and diabetes greatly increase the risk of pancreatic cancer. So to compare with someone who does not have either of these conditions, the risk is 34-fold increase. Compared to type 2 diabetes, CP diabetes, the risk is 12-fold increase. Furthermore, CP diabetes has been associated with a 30 to 50% higher risk of all-cause mortality in multiple studies compared to type 2 diabetes. Renal and cardiovascular deaths appear to be similar, but there are higher rates of death due to respiratory, infectious, and GI causes. And cancer mortality is higher in women with CP diabetes. In addition to the mortality concerns, there are also additional morbidities. So CP diabetes versus type 2 have higher rates of hospitalization for various GI causes, chronic pulmonary disease, renal disease, and infectious disease. So clearly what is needed are biomarkers to allow us to distinguish CP diabetes from type 2 diabetes. In terms of prediction of future diabetes in patients with CP, we've started to look at this in our studies. Let me show you studies today from the PREDICT-3C study, which is conducted in a cohort called PROCEED, where 645 participants have confirmed chronic pancreatitis. And of those, 276 had CP diabetes. In this study, we analyzed multiple clinical features in univariate and multivariable models to build prediction models for diabetes. We also have a poster on the study at this meeting where you can see the results of analyses where we stratify the diabetes group based on the timing of their diabetes diagnosis in relation to the pancreatitis diagnosis. In univariate analyses, we found a number of features significantly associated with diabetes and CP. These are listed in the left column. They include things such as age, BMI, sex, family history of diabetes, pancreatic calcification. The middle column shows features that were borderline associated with diabetes and CP. And the final column shows features that were not significantly associated. So what we then did was we took the significant and borderline significant features and analyzed them jointly in a multivariable analysis. And interestingly, even when analyzed jointly, many of these features remain significant. So I've highlighted in yellow on the top features that we consider to be classical type 2 diabetes risk factors, such as age, BMI, being male, non-white races, and tobacco use. Other significant features highlighted in blue are more reflective of pancreatic damage. So including a history of AP complications. Interestingly, alcoholic etiology was protective. This could also mean that non-alcoholic etiology increases the risk. Exocrine pancreatic dysfunction, pancreatic calcification, and pancreatic atrophy were also associated with diabetes and CP. These features are summarized here with their odds ratios. So what we find is that not only do classical type 2 diabetes features, but also pancreas damage-related features predict diabetes and CP. And this could be that risk factors for diabetes may accelerate pancreagenic diabetes, or this may represent a double hit. You may have someone who's destined to develop type 2 diabetes, perhaps based on their insulin resistance and their genetics, and then the pancreatic damage accelerates that process. Or if CP diabetes is a distinct condition, it may be that some patients have both. Thinking about chronic pancreatitis and diabetes, I certainly remember before I started in this field, I thought it was a no-brainer. Chronic pancreatitis, your pancreas is scarred and destroyed, and so your beta cells are destroyed. What is there to be interested in? It turns out that's actually not the case. We see glycemic dysfunction, beta cell dysfunction well before the end stage fibrotic phase of chronic pancreatitis. So it's actually a functional deficit and much more interesting than simply a destructive process. So if we look at the scheme here, we go from AP to recurrent acute pancreatitis to CP, inflammatory phases in the middle where beta cells they're still there, but they're dysfunctional. And then with time, destruction of the beta cells. So in this model, we would expect worsening insulin secretion and increased rate of diabetes over time. And indeed, this has been observed. In several studies, here are a couple where we see that at diagnosis, 4 to 10% have diabetes. 1 year out from chronic pancreatitis, that goes up to about 8%. 10 years into CP, 30 to 50% develop diabetes. And by 20 plus years, the majority of patients with CP will have developed diabetes. A meta-analysis of 15 studies supports this as well. They found that the overall incidence of diabetes in CP was 30%, but the main risk factor that predicted this was actually duration after diagnosis, with higher rates with increasing duration. Well, the insulin deficiency or beta cell dysfunction is often the first thing we think about, not surprising given the location of the beta cells. However, other features may play a role in the pathophysiology. Several studies, including detailed clamp studies, have actually documented insulin resistance in patients with CP diabetes. Hepatic insulin resistance may be a feature related to deficiency of pancreatic polypeptide. And in fact, there there have been some studies where they administered pancreatic polypeptide and then improved glucose tolerance in patients with CP diabetes. So there have been some proposals that deficient pancreatic polypeptide response to mixed meal stimulus may be a biomarker for pancreatogenic diabetes, also known as type 3C diabetes. So we're taking a close look at this in a study we call DETECT, where we're administering meal tolerance tests to patients with type 2 diabetes and two forms of pancreatogenic diabetes, that associated with pancreatic cancer and that associated with CP. And we draw multiple time points after the ingestion of the liquid meal. What we have found, indeed, here I'm showing you fasting results where the pancreatic polypeptide concentration was lower in both groups, and it was actually lowest in CP diabetes, that's on the far right, compared to the type 2 diabetes in yellow on the left. And then looking at throughout the meal tolerance test, the area under the curve of the pancreatic polypeptide response is quite low in chronic pancreatitis-related diabetes. It's on the bottom there. Pancreatic cancer-related diabetes in the middle, and the highest response is seen in type 2 diabetes. Another potential player in the pathophysiology of diabetes and CP is the incretin system. So incretins are GLP-1 and GIP. These are hormones secreted by the gut in response to nutrient ingestion. It's a signal that enters your bloodstream and tells the pancreas to make insulin. As you know, exocrine insufficiency is common in CP. So there's a hypothesis that this impaired nutrient digestion may lead to a reduced incretin response, which would then contribute to postprandial hyperglycemia. And indeed, this system has been found to be impaired in type 2 diabetes. Studies so far are inconclusive in CP diabetes. Now, this is important for the treatment implication. For type 2 diabetes, we have GLP agonists and DPP-4 inhibitors. However, few of us would feel comfortable giving that to pancreatogenic diabetes because both of these agents have been linked to pancreatitis itself. So what can we do as far as helping these patients with their incretin system? There have been a few small studies actually where they assessed the effect of giving pancreatic enzyme replacement on the incretin system and glycemia. Two of these were in CP, two were in cystic fibrosis. And what they found, while effects on glycemia were inconsistent, there did seem to be an increased incretin response related to administering pancreatic enzymes. So this will require further study. So to close, we still are not certain whether CP diabetes is a distinct form or a subtype or an accelerated form of type 2 diabetes. We're still looking for biomarkers to distinguish these two forms of diabetes. And it may be that a multifactorial model may be needed to distinguish the two. Blunted pancreatic polypeptide response is a feature of CP diabetes, but the overlap precludes its use as a diagnostic biomarker at this time. So future investigations will be needed to further understand the mechanisms underlying CP-related diabetes. And this will then inform treatment trials, of which at the current time there are very, very few, leaving those of us taking care of these patients to essentially guess at how to treat them. Thanks for your attention.
Thank you, Mark, for the excellent presentation. Our next speaker is going to be Dr. Jason Bruce, who's a senior lecturer at the University of Manchester. He did his undergrad and PhD from the University of Manchester, and then came to the University of Rochester for a postdoc research associate position, after which he went back to Manchester. He also did a sabbatical subsequently in Dr. John Williams' lab. His lab focuses on projects relating to pancreatitis and pancreatic cancer, specifically the role of metabolism and cytosolic calcium homeostasis. His lab is also interested in the protective effect of insulin, on acute pancreatitis, which is going to be the focus of his talk today, which is titled Role of Insulin in Pancreatitis.
Okay, so good morning, everybody. I'd like to thank the organizers for the kind invitation and for organizing such a fantastic conference. So first of all, I have no conflicts of interest. So today I'm going to be talking about the protective role of insulin during acute pancreatitis. And I'm going to talk about two different stories. And first of all, hopefully going to convince you that insulin has a direct protective effect against acinar cell injury during acute pancreatitis, but also we've got a new story that insulin regulates the expression and secretion of antimicrobial peptides from pancreatic acinar cells into the gut. And we think that both of these phenomena are important for preventing the progression from mild to severe acute pancreatitis. So this slide kind of illustrates many years of pioneering work by many different groups. And it shows that the major causative factors include excessive alcohol consumption and fat consumption and bile acid reflux from gallstones. And all of these factors, all of these agents can converge on the mitochondria to impair metabolism, leading to ATP depletion and a cytotoxic calcium overload response. Both of these then contribute to necrotic cell death and a spiral of self-perpetuating tissue injury, which leads to the systemic inflammatory response. What I hope to convince you is that insulin can have a protective effect on many of these different stages. So, as many of you know, there's a strong link between pancreatitis and diabetes, not least because pancreatic injury will lead to collateral injury of the pancreatic beta cells, leading to impaired insulin secretion. And that is described as type 3C diabetes, as described in the previous talk. But there's also evidence that pre-existing diabetes can make pancreatitis more severe. And there are many potential mechanisms for why this could occur, particularly systemic effects of a loss of insulin and hyperglycemia, which will all contribute to a more severe pancreatitis. And it was a few years ago when we first demonstrated that insulin directly protects acinar cells, and it does this by preserving cellular ATP and maintaining a low resting calcium. So therefore, if there's a loss of insulin, you effectively lose this protective mechanism, and this exacerbates the cellular injury, which then contributes to the severity of the disease. So we first examined this using two different experimental models of pancreatitis, the classical cerulean hyperstimulation model, and then the more recently described palmitoleic acid and ethanol model that was first described by Robert Sutton and David Criddle a few years ago, and has been characterized by other groups. And we did these experiments on the type 1 diabetic Akita mouse and a pancreatic acinar cell conditional insulin receptor knockout mouse that was generated by John Williams and Dolores Sands at the University of Michigan. And I'm going to whiz through these data very quickly because we've published these data. These were published last year in Nature Communications. And what this slide illustrates is that cerulean-induced pancreatitis is more severe in diabetic mice. So this just shows the histology injury score. And the take-home message from this is that endogenous insulin is protective during acute pancreatitis. However, it's very difficult to separate the systemic effects of a loss of insulin and hyperglycemia, which makes pancreatitis worse, from a direct effect of insulin on the acinar cells. And the only true way of examining this is if we knock out the insulin receptors within the acinar cells selectively. And these mice were generated by John Williams by crossing the elastase-Cree ER mouse that was generated by Craig Logston with the double-floxed insulin receptor mouse, which was developed by Ron Kahn. And this means that the Cree recombinase insulin receptor fusion protein is expressed within the acinar cells, but remains inactive until we feed the animals tamoxifen, which then deletes the insulin receptor specifically in the acinar cells, but importantly in adult mice, because insulin is required for the normal development of the pancreas. And as you can see, these mice are normoglycemic. They secrete insulin normally. They are not diabetic. They simply don't express the insulin receptor within the acinar cells of the pancreas. And all of the tissues, such as the spleen, liver, muscle, kidney, gut, heart, brain, you name it, all express the insulin receptor normally. And again, I'm just going to whiz through the data very quickly because these have been published, but the take-home message here is that cerulean-induced acute pancreatitis is more severe in Perserco mice in which the insulin receptor has been deleted. And this just shows the histology injury scores. And again, palmitoleic acid and ethanol-induced pancreatitis, which is a much more etiologically relevant model of pancreatitis, is also more severe in the Pacerco mice. And again, here's the histology injury scores. So the take-home message so far is that endogenous insulin directly protects pancreatic acinar cells during acute pancreatitis. If this is true, then one might expect exogenously applied insulin may also have some therapeutic utility. And we tested this using the hyperinsulinemic euglycemic clamp, in which we can infuse cerulean to induce pancreatitis, but then also infuse high-dose insulin. So this dose, which you can see, 12 milliunits kilogram per minute, is about 3 times higher than what the pancreas would, what the animals would ever see. And we can monitor glucose every 15 minutes and then infuse glucose to maintain a very tight glucose range within the physiological range. And then we measured plasma amylase as a readout, an early readout of acute pancreatic injury. And as you can see, when we infuse cerulean over just 5 hours, we see a 3-fold increase in plasma amylase, suggesting that these mice have the early stages of acute pancreatitis. And importantly, when we infuse the insulin 2 hours into the cerulean infusion, so this is mimicking therapeutic intervention, there is a significant reduction in plasma amylase. So this gives us this tantalizing possibility that insulin infusion could be therapeutically beneficial, at least in mice. Whether this could be translated to severe acute pancreatitis patients that are on critical care is a very different story, and I'd be happy to discuss this later. So in cellular models, the mechanism for insulin's protection, we can show that insulin prevents palmitoleic acid-induced ATP depletion, inhibition of the plasma membrane calcium ATPase, which is the mechanism that maintains low resting calcium in the acinar cells. It also protects against cytotoxic calcium overload and necrotic cell death. And all of these effects are prevented in the Pacerco mice, in which the insulin receptor has been deleted. The mechanism for this involves a metabolic switch from mitochondrial metabolism towards glycolysis, very similar to what we might observe in cancer cells. This is effectively like a short-term Warburg effect. And we assess this by measuring NADH autofluorescence, and NADH is mainly produced by the Krebs cycle within the mitochondria. But if we inhibit the mitochondria with a protonophore, CCCP, you get a drop in NADH, which represents mitochondrial metabolism. But there's also another source of NADH, which is the glycolytic enzyme glyceraldehyde phosphate dehydrogenase. And if we inhibit that enzyme with iodoacetic acid, you get a further reduction in NADH autofluorescence. So what we've got there is a measure of glycolysis. And when we then repeat this experiment in the presence of insulin, you can see there's this switch from mitochondrial metabolism towards glycolysis. And the molecular mechanism for this insulin protection, we have also confirmed, is due to AKT-mediated phosphorylation of the key glycolytic enzyme, phosphofructokinase, fructose bisphosphatase 2. Now, this is a very important regulatory mechanism for driving glycolytic flux, because fructose 2,6-bisphosphate is an allosteric activator of the rate-limiting step of glycolysis, which is PFK1. So this is acting, and in particular, this enzyme, PFKFB2, is a tunable enzyme. And so when it's phosphorylated, its kinase activity is increased. So this is acting like a volume control on glycolysis to preserve cellular ATP and therefore maintain the calcium pumps and any other ATP-dependent process for that matter. So to summarize this story so far, we have shown that insulin directly protects against acinar cells by increasing glycolytic energy supply. And this might explain why pre-existing diabetes, pancreatitis is more severe, but I would go one step further and hypothesize that the transition from mild to severe acute pancreatitis depends on the extent of the collateral beta cell injury, and therefore intervening early enough with either insulin or an insulin mimetic that might mimic some of the downstream signaling pathways could prevent this transition towards a severe phenotype. Okay, so in addition to a direct effect against acinar cell injury, insulin also regulates the expression and secretion of an antimicrobial peptide called Reg3 beta. And we believe that this maintains a healthy gut microbiome, it maintains a healthy gut barrier function. And during diabetes, due to a loss of insulin, there's a loss of Reg3 beta within the pancreas and a loss of Reg3 beta secretion into the gut. This leads to gut dysbiosis, a loss of gut barrier function, leading to bacterial translocation, infected pancreatic necrosis, and a more severe acute pancreatitis. We know that Reg3 beta has antimicrobial properties, as shown by this E. coli growth colony. We also have shown that Reg3 beta is almost completely depleted within the pancreatic tissue. If we take the acinar cells, stimulate them with CCK so that they secrete, and then take the supernatant, filter it, and then run it on a Western blot gel, we can see Reg3 beta in the supernatant, so it's secreted from the acinar cells. If we then take this supernatant and then add it to the bacteria, we can see a reduction in the growth of the bacteria, but this is abolished from the Pacerco mice. So clearly, whatever's being secreted from the acinar cells has antimicrobial properties, but this is lost in the Pacerco mice. We've, in addition to a loss of Reg3 beta from the pancreas tissue, there's also a loss from the fecal samples. And so this suggests that pancreatic acinar cells not only secrete Reg3 beta, but secrete Reg3 beta into the gut. The other important point here is that this suggests the extent of this depletion. It suggests that pancreatic Reg3 beta may be a major source of gut Reg3 beta, which flies in the face of the dogma that the Paneth cells of the gut are the major source of these antimicrobial peptides. The other important point is that the size of Reg3 beta is much smaller in the fecal samples, and the reason for this is that it gets cleaved by trypsin that's co-released by the acinar cells. So this is essentially a similar mechanism to what happens with zymogens within the gut. So Reg3 beta is being secreted and then activated within the gut, where it increases its antimicrobial properties. If this is true, then one might expect that the Pacerco mice will exhibit a gut phenotype, and that is what we see. So first of all, we see an elevation of fecal calprotectin, which is the clinical biomarker for inflammatory bowel disease. We also can detect CD45 in Pacerco small intestine and colon. We see a reduction in goblet cell number in the colon, and these are the cells that secrete mucus. And then finally, we also can detect bacteria using FISH, and we see adherent bacteria within the colonic crypts, suggesting that the bacteria have breached the mucus barrier layer and are now binding to the epithelial cells and possibly translocating into the blood. So to summarize then, insulin regulates Reg3 beta expression and secretion into the gut, where it maintains a healthy gut microbiome and barrier function. But when during diabetes, the Reg3 beta secretion is lost, leading to dysbiosis, inflammation, bacterial translocation, and possibly a more severe acute pancreatitis. It just leaves me to thank the people that did the work, Rosa and Sarah, who were funded by an MRC grant, but also none of this work could have been done if it wasn't for a very long-standing and important collaboration with John Williams and Dolores Sands. And John is retiring in the next few weeks, and I think it's important to emphasize the Herculean contribution that he has made to the pancreatology field. So thank you very much for your attention.
Thank you very much. That was very, very exciting research and recognition of Dr. Williams, who we all know for a long time. Our next speaker will be Dr. Melina Bellin. And Dr. Bellin is a physician scientist and professor in the Department of Pediatrics, Division of Endocrinology, and Department of Surgery. Dr. Bellin co-chairs the T1D-APC consortium, and she is a leader in diabetes management, pancreatitis management, and TPIAT and its outcomes. And we're just very excited to have her speak today and give us more enlightenment and update us on islet autotransplantation. Dr. Bellin.
Thank you. Good morning, everyone. Happy to be here talking on updates in TPIT. I have some disclosures, but none of this is directly pertinent to my talk today. In the scope of this 12-minute talk, I had to pick a few things to focus on, and I'm going to tell you mostly a story about diabetes and endocrine results after TPIT, given the session here today. But I broke this apart into 4 parts. I think many people here are familiar with TPIT, but I'll give you a little background on the surgery, particularly with what we know about surgical approaches here in the US from our post TPIT registry. I'm then going to talk a little bit about the early biomarkers for diabetes risk and our diabetes outcomes and how we're trying to address this with clinical trials. I'll then talk about long-term outcomes, especially as it pertains to diabetes, and some of the nutritional implications of TPIT that I think will actually overlap quite nicely with some of Dr. Hart's talk from earlier today. So total pancreatectomy with islet auto transplant, which I'll have in these slides abbreviated as TPIT, of course, is a surgical procedure that is used to treat patients who have fairly clinically severe forms of recurrent acute or chronic pancreatitis. And the goal of doing this surgery is really to provide pain relief and improved quality of life for these patients. That's a really important part of the surgery, so I apologize that I'm not going to talk much about that. It's not because it's not important, but I am going to focus a little bit more on the islet auto transplant part here. And the islet auto transplant is really an attempt to prevent or minimize the severity of surgical diabetes when you perform a total pancreatectomy to treat pancreatitis. Most of the patients we see for this procedure are quote unquote early enough in the course of their disease process that they don't yet have diabetes, but we do see about 8 to 10% of patients who already have more mild early diabetes from their chronic pancreatitis. So when we resect the pancreas, of course, there is additional anatomic considerations here. And so we looked at this from the post-study, both in adults and children. Most of our surgeries are open across the US, but there are some laparoscopic assisted approaches and about 1/4 of patients and a few that are fully laparoscopic. Most patients have splenectomy at the time of TPIT, and in most cases, the pylorus is preserved, but this is a little bit more variable for adult patients than children, and most commonly GI reconstruction with a Roux-en-Y procedure, although again, a lot more variability in the spectrum of surgical approach and intestinal resection reanastomosis in the adult population compared to the pediatric population, and choledochojejunectomy is the most common approach for biliary reconstruction. Most patients, so there's a gastroparesis that happens at least transiently and sometimes long-term after this surgery. In order to manage nutrition after the surgery, most patients, but not all, have an enteral feeding tube placed. We do see one of the complications of islet infusion being a partial portal vein thrombosis. We give anticoagulation to reduce this risk. But if we look at this risk in adults and children, it's much more common in the adult population than the pediatric population. So looking at diabetes risk, one of the things that our patients always ask us when they come in for this surgery is, what is my chance I'm going to have to stay on insulin after surgery? We don't necessarily select candidates for surgery based on their likelihood of coming off insulin. That's really based on pain control, but patients would like to know what their prognosis is. One of our visiting surgeons from Japan, Yoshi Nano, looked at this in our population, about 370 of our patients, in terms of insulin independence and measures that we could obtain before they had surgery and how predictive that was of outcomes for insulin independence. Patients who are younger, and we know this about pediatric, but this is also true of young adult patients. Patients who are younger in general are more likely to have insulin independence, particularly as you get out longer from transplant at 5 years. Having a more normal body weight is favorable, and having lower glucose measures of A1C, fasting glucose, and 1-hour stimulated C-peptide convey more favorable insulin independence. And there is also a relationship with fasting C-peptide. So this is really, really strong at a population level, as you can see from the p-values in this kind of busy table here. But if you put all of these into a multivariate analysis and try to look for any individual patient using that logistic regression modeling, we actually only predict correctly about 70% of the time. So even using the best data we have, about 1/3 of the time, we would predict an error whether someone was going to be on or off insulin after surgery. So I say this just to say that we have a lot of population level associations. We're not as great as I would like to be at being able to tell an individual patient what their outcomes are. There are a few exceptions though. If someone has impaired glucose tolerance before they go into surgery, if they have frank diabetes, though a pre-diabetes A1C, those are poor prognostic factors. I usually tell those patients to expect to remain on insulin. The other thing that actually is out of a separate paper here is if we look at islet autoantibodies, most of the time they're negative, but we have a few patients who have positive type 1 diabetes autoantibody markers, and they almost always confer a poor, I shouldn't say a poor diabetes outcome. This doesn't mean they're poorly controlled, but a poor islet graft function outcome. So one of the reasons I think that we're not that great of predicting is because even patients who have a good islet yield and a lot of islets transplanted, we know that we lose for various reasons many of those islets as part of the transplant process. This is an opportunity for us, however, I think to improve. So optimistically, maybe something we can do better, but I think this explains a great deal of the variability. We've known this for a long time from animal models of islet transplant, where we can actually do histology. We obviously don't take out someone's liver after we infuse islets in the liver. So it's hard to know specifically in people how many of these cells survive. But in the type 1 diabetes world, there are now beta cell death biomarkers that have been used heavily in type 1 diabetes research. And this graphic that I'm showing you on the slide is a particular marker called unmethylated insulin DNA that is specific to the beta cell. And these are circulating serum measures right before and then after TPIAT. And the one thing we see is that in all of the patients where we measure this, we see elevation of this beta cell death marker. So we do know that, like the animal studies, we're losing some islets when we transplant these patients. So the question is, how can we address that? And so some of the areas that we're looking at right now, there's an early inflammatory response that occurs when we infuse the islets called IBMIR, or instant blood-mediated inflammatory response. So that's one of the targets that's been investigated in this field to try to improve islet engraftment. We also think that there is a beta cell stress factor that particularly can be exacerbated by hyperglycemia. We don't have as much to directly test to address that, but we are measuring kind of biomarkers of that as well. So one of our, we've done several trials in this area of trying to improve early islet graft survival, and particularly targeting that inflammatory response. One of our recent trials was looking at TNF alpha blockade, which has shown some promise in non-controlled studies, as well as alpha-1 antitrypsin that actually had some really good non-human primate data to back that. We were somewhat optimistic when we looked at a number of markers of insulin or islet function and insulin production at 3 months. And this is one of them, called the acute C-peptide response to glucose. So it's a measure of a kind of a sensitive measure of islet health, where you give a glucose bolus or a dextrose bolus and measure insulin secretion. And at 3 months after transplant, so we had patients randomized to either no treatment, etanercept, or alpha-1 antitrypsin. We saw a signal that the treated patients, particularly those with TNF alpha blockade, perhaps were doing better. Unfortunately, when we look at the 1-year data, and we have 2-year data that we'll have shortly, this doesn't appear to be sustained. So it may not be potent enough to change the long-term outcomes, and it didn't change insulin independence. We've looked at several other drug therapies and diabetes technology, and Dr. Bielman, who's in the audience, is also looking at some surgical approach to address this, which I won't get into here. We've also looked at measures of insulin misfolding and islet amyloid misfolding. And basically what you see here in the graphics is ratios of beta cell hormones, or islet proinsulin hormone to C-peptide ratio, and pro-islet amyloid polypeptide precursor to the formed product. So these are just basically elevated numbers on this graphic are just measures of ER stress and protein misfolding in beta cells. And when we look at these at 3 months after TPIAT, the patients who become insulin independent have much lower ratios, meaning they don't have as much misfolding and ER stress present as the ones who remain insulin dependent. Now, we don't have a drug therapy that directly targets this, but we do know from populations like type 2 diabetes that hyperglycemia is probably one factor that can exacerbate this ER stress pathway by inducing more insulin secretion. So this is part of our rationale for really trying to maintain tight glycemic control. So looking at long-term outcomes after TPIT, we do see sustained pain relief for up to 10 years. And we have an abstract here that I'll point you to tomorrow with one of our medical students looking at patient survival. In terms of islet graft function, we see some patients who maintain insulin independence for more than 10 years. And in fact, in our longest case, we have someone who is actively off insulin 22 years after surgery, and a child who is no longer a child, is a young adult, but is off insulin 18 years after surgery. So although there is an attrition of islet graft function and insulin independence over time, we do know that it can be maintained long-term. And we think that maintaining islet function is beneficial to these patients. And so first of all, by maintaining islet function, we get tighter glycemic control. We see that reflected in patients who are insulin independent, having an A1C below 6, and then patients who just have a functioning islet graft, having a A1C on average below 7 versus those who have graft failure. The other thing that we've seen is we collect quality of life data prospectively, and those patients who are insulin independent long-term, and this is based on over 500 patients with more than 2,000 time points of follow-up total, that those patients who are insulin independent have higher quality of life scores on their, this is an SF-36 in this case, and I won't go through the adjusted models, but this is true even if we adjust for pain, opioid use, age of the patient, things that you would think would affect quality of life. So we would like to study this further. We have an ongoing trial right now funded by NIH that we call LIFT, and it's long-term islet function after TPIT. We have right now very gross clinical data, and what we want to get is really detailed granular data on islet physiology and try to translate that into benefit on health and quality of life, including diabetes complications. And we think this is important because there are some populations that cannot access TPIT because of insurance. So finally, just a couple last slides to round it out on the nutritional implications after TPIT. You heard a lot about this as for chronic pancreatitis from Dr. Hart, but these patients, much like the chronic pancreatitis population, are at risk for fat-soluble vitamin deficiency. And we demonstrated that initially in our pediatric population at the University of Minnesota. But Dr. Downs, one of our pediatric GI docs, worked on the post data with Dr. Maisam Abu Al-Haija and me, and she's going to present that data on fat-soluble vitamin deficiencies actually later today. One of the obvious consequences of fat soluble vitamin deficiency is bone health issues. And so when we look at bone health before and after surgery, one thing we do see is in the 1 year after surgery, we see a significantly decreased bone mineral density, particularly in the hips. So this is this 3 to 4% decrease in bone mineral density at the hips is greater than what you would expect in the average population. And it is accompanied by markers of bone turnover, so an elevation in serum CTX. So we do think right in this postoperative period that there's some bone health, some bone turnover that needs to be addressed. It does appear, though, optimistically that decline slows after 1 year. And in fact, if we look from 1 year to 18 months, so this was data we had from a clinical trial, it was very stable. But we do recommend that these patients get routine DEXA scans. I don't think we know the correct interval, but are monitored after surgery. So I just want to acknowledge our whole team who has contributed to this, the members of the POST consortium, and a number of our trainees and fellows who are acknowledged there, as well as our funders. Thank you.
Thank you, Melina, for an outstanding presentation. I'll ask all the speakers to come up to the podium. And I hope that Dr. Goodarzi is available online to answer any questions. So we'll take about 5 to 7 minutes of questions. So Hel?
Thank you. Excellent session. The question is focused. Sorry, can you hear me now? The excellent session. The question is focused towards Dr. Jason Bruce. Fascinating work on 3 lines of evidence. I'll keep this short, but 3 lines of evidence. It's so fascinating because of all the clinical interest in diabetes. I saw patients with DKA who came in with pancreatitis, and many of us do. The second is that in many proteomics, including today at a poster session, there are reduced proteins involved in bioenergetics that are in line with your pathway for insulin. But the third, which is the question I have for you, is that about 20 years ago, I remember, looking at just experimental severe pancreatitis histology and noticing that the peri-islet Acinar cells are relatively spared compared to the rest of the Acinar cells.
Yeah.
And so the question is there a paracrine effect of insulin or other factors from the islets that could be a holy grail in protecting the Acinar cells in the setting of inflammation?
I think the answer to that is yes. No, I mean, there's a lot of anecdotal evidence from histology dating back, many years. And interestingly, the guy behind you, Peter Heggie, actually reported this in a paper a number of years ago, where he showed that the Acinar cells were relatively intact during several different models of acute pancreatitis. So I think that kind of speaks for itself, really, that, yeah.
I wanted to see this picture on there.
Yeah. Okay. So I think, yes, it's a very good point. Yeah.
Two pointed questions. The first to Dr. Hart. Do we have clinical evidence once identified bone demineralization in chronic pancreatitis that we can successfully treat it? And I'll go ahead and just state my second question. Is Dr. Bruce, do we have evidence that the Reg3B is present in humans and has the same functionality as mice? Because I don't think any of your data was human data, if I remembered it correctly. Thank you.
So just to clarify, the question was, do we have
Yeah,
clinical
do we
evidence
have
of-
evidence that once you've identified bone demineralization in chronic pancreatitis, we can successfully treat it?
No.
Good.
Not that I'm aware of.
Any thoughts about how we can improve that?
Yeah, lots of options. I mean, there's, the sort of standard approach, right? And the general population is vitamin D, calcium supplementation. If it's not already met by just routine dietary intake, smoking cessation, if that's a factor, weight-bearing exercise, and then in patients with, high risk for fracture, additional medical therapies. There are also some data, it's observational only, that suggests that there may be a role for pancreatic enzyme replacement therapy. I mean, obviously in those patients who have known or established extreme pancreatic insufficiency, that's a no-brainer, but perhaps even for those without, overt steatorrhea. And then otherwise, if we think that mechanism, which I believe that inflammation is a huge contributor to the loss of, bone mineral density, somehow interrupting that would and should, I think, improve bone health, as well as many other things. Dr. Goodarzi talked about the diabetes aspect of things, for example.
One comment on that also. One comment on it also, if you look at Phil's paper, he talked about premenopausal women and men that have chronic pancreatitis. But the other thing that he reports is there's an age shift also. So mechanistically, something is going on that we don't know. It's not necessarily duration of disease. I don't really know for sure, but there's an age shift when you look at the graphs in people who have chronic pancreatitis that have osteopathy.
If I could just answer Fred's question about Reg3 beta. So it's a very good question. Reg3 beta is mouse specific. It's not expressed in humans, which may raise the question, well, what the hell, is this really relevant? There is a lot of homology with some of the human Reg3 related C-type lectins. Most notably Reg3 gamma, which is mouse, but it's probably the most important one that's related to humans. And unfortunately, I didn't have the time to show you, but that is also depleted in our Pacerco mice, both in the pancreas and in the gut. We've also looked for another important antimicrobial protein, which is glycoprotein 2, which is one of the most abundant zymogen granule proteins. And this also has antimicrobial properties, and that's also depleted in the pancreas of our Pacerco mice. Okay.
So Peter, just one second, we'll go to this side for a question, please.
Yeah, hi, just to switch to auto-islet transplantation. So you show the graph of the attrition of insulin producing cells. I had a question about glucagon. So if you completely remove the pancreas, you don't have a good source of glucagon, and then you take insulin and you become hypoglycemic. And so is there any evidence that glucagon production is maybe a little better preserved than insulin production?
Right. So I did not show the glucagon data, but we do have data on glucagon, and we're actually looking at this in a little bit more detail because the data we have is largely from insulin independent patients in the past. If you stimulate them with arginine, which stimulates both insulin and glucagon secretion, they have a pretty normal glucagon response compared to a healthy control patient. If they have all of their islets in the liver and you put them in a hypoglycemic clamp situation, their glucagon response is less than a healthy control. If you, however, have someone who has part of their islets in the liver and part of their islets outside of the liver, usually in the peritoneal cavity, they have a better glucagon response than when all islets in the liver. So we think that there is, the theory is that there's some glycogen flux in the liver that actually messes up to some extent the normal physiologic response to hypoglycemia, and that is a limitation.
And is there any evidence that those cells are better preserved over time? Like, even if you don't have enough insulin, at least you don't have brittle diabetes.
Right. And that's what we believe. I mean, we see our patients, in clinic who have this, and if they have islet graft function, a lot of them are, you can put a CGM on them and they'll be 70 to 180, 95% of the time, even though they're on insulin, which is way better than a patient with type 1 diabetes. Now, I've tried to convince Medicare of that. They don't believe this yet, but I do think that having functioning islets is much better than just being completely on insulin.
Peter.
So thank you very much for this session. This was absolutely fascinating. So I would have two questions for two persons. One of them would towards to Mark. Actually, we see plenty of times in the unit when a patient's coming with slight obesity, with actually oral anti-diabetic treatment, with very mild diabetes around like 6-ish hemoglobin A1C. During pancreatitis, there's a necrosis and we need to start insulin. So when to reconsider the treatment after AP, because we know that insulin has a trophic effect, as Jason showed, maybe had the pancreas to regenerate, but of course it's against the obesity and all the other part. So when we send the patients home after, how to put them on treatment and when to reconsider about the anti-diabetic treatment of this patient?
That's a great question. And as I mentioned, there really has not been extensive clinical trials or study as to how to treat these patients versus just diabetes in general. So I think at the current time, we would treat the diabetes as we would, anyone who's coming into the hospital with diabetes. If it's severe, if they've come in with DKA, they may need insulin, and they may not need to be on insulin long term. They're of course, once they get home, often dictates that. And of course, knowing what they were before they came into the hospital is often very helpful. So we always look at the A1C on admission, and if it was, really, really high, and we're on oral agents, that might not be a good regimen for them. But we really don't have specific recommendations at this time for the pancreatitis patients with diabetes.
Okay, thank you. And I have questions to Jason. This was very fascinating. So thank you for mentioning this issue. I always want to see the ring as well, but never mind. So actually, this is a very good concept that pancreas is an antimicrobial organ. And this hasn't been discussed details. I know that Shmuel Mualem had a very elegant study concerning the OROI1 channel mediated antimicrobial secretion of SNR cells. So how is your model? How insulin? Is it insulin playing via ORI1 issues or not? This would be my first question.
Well, the work of Shmuel's was basically the inspiration for our work, really. And so he showed that cramp, cathelicidin-related antimicrobial peptide, was likely the most likely antimicrobial peptide to be secreted by the Acinar cells. And I think with the ORI1 knockout, you're basically knocking out secretion entirely in the animal. And so everything will be lost. So that's why it was such a profound effect on the gut phenotype. Now, we've looked for cramp and we couldn't detect it in the pancreatic acinar cells. And if we did a Western blot with the pancreas tissue, occasionally we could detect it, but whenever we isolated the acinar cells, we couldn't detect cramp. Then we did some immunohistochemistry and we could detect cramp within the islets. So we think that cramp is important in islet function, perhaps the alpha cells, the beta cells, we don't really know. It's hard to distinguish, but we don't think that cramp is secreted from the acinar cells. And so in terms of Reg3 beta, its expression is regulated by insulin. And therefore, if you deplete the insulin, you deplete Reg3 beta and you lose that antimicrobial secretion. So it's an insulin-dependent antimicrobial secretion from the pancreas that is relevant in terms of the severity of the disease, in my opinion.
Thank you. And one last question.
No, we're gonna have to stop. So let's just take the rest of it offline here. This is the unbelievable session. Everyone's very interested in this interaction between diabetes and chronic pancreatitis and beta cells and all this wonderful stuff. So, but we're going to need to move on to our next important event, which is our Frank Burt's lecture. Congratulations and thank you all very much.